And now I introduce ronnen Beeri, Professor Ronnen Beeri from Hadassah. I was grateful to spend with him 2 years in Boston. And he's going to speak about the mitral valve and the left ventricle. It takes two to tango.
Good afternoon, and again, I thank the organizing committee for inviting me to give this tribute to my mentor. And I want to expand a little bit on what Ehud has just presented to you and speak a little bit about the interplay between the left ventricle and the mitral regurgitation and how they influence one another. And as you've seen, in situation in which you have an inferior-posterior MI, the posterolateral papillary muscle gets tethered, and you can see that that induces incomplete closure and mitral regurgitation. And actually, as Chaim Yosefi has shown in a paper in Circulation 2 years ago, whenever you have also involvement of all four apical segments, the same mechanism applies and you get mitral regurgitation. And we know that after a myocardial infarction, expansion of the infarcted tissue begins almost immediately. And this process, which we call remodeling, is actually a compensatory mechanism at the beginning, but subsequently becomes maladaptive, and it generates a spherical ventricle which does not contract. We also know from epidemiological work, quite old ones, that even the mere presence of mitral regurgitation doubles mortality following myocardial infarction. But how does one mitral regurgitation affect remodeling and vice versa has not yet been assessed in a controlled manner. So Bob had the idea that this vicious cycle is something that is basically driven by two forces. The one is the ischemia, or the infarction, that actually decreases contractile function and induces LV dilatation. That LV dilatation induces a rise in wall stresses, and mitral regurgitation actually increases these wall stresses, further decreasing contractile function. And this goes on by inducing hypertrophy, eccentric hypertrophy, which when it becomes maladaptive, when the cells have been driven into a division they cannot complete, induce failure and apoptosis of those cells. Additionally, extracellular matrix remodeling occurs, and the ventricle becomes eventually enlarged and malcontracting. We have not yet even addressed the influence of different paracrine and endocrine factors that are released during a situation in which the heart is stressed and influence every step of this vicious cycle. To address this subject, Bob, along with our very gifted animal surgeon, Luis Guerrero, and myself, we devised a model in which we tried to separate the two elements of mitral regurgitation and myocardial infarction. And basically what we did, we implanted a shunt between the lateral wall of the left ventricle and the left atrial appendage, which induced a fixed MR-like flow independent of the valve itself. In this model, we induced also a myocardial infarction, a small apical myocardial infarction by LAD ligation, which does not induce by itself mitral regurgitation, native mitral regurgitation. We did that in a sheep model. The regurgitant fractions as calculated both by flow model and transonic flow meters were around 30%, equivalent to a moderate mitral regurgitation in a clinical setting. That shunt that I've described to you has also the additional advantage of being amenable to simulation of repair. So actually you can easily suture the shunt and simulate mitral valve repair and check the effect of that. So in the first experiment that we did, we tried to first answer the question whether remodeling and ischemic MR are potentiating one another. So we had two groups of sheep. One of them had a myocardial infarction with a shunt implanted, and the other group had the shunt ligated at the outset, simulating, it's a sham operation, so it's only MI. And as you can see in this slide, both volumes were significantly larger, both end-diastolic and end-systolic volumes, and contractility, as measured by invasive hemodynamics using a Millar catheter, both DP to DT and both preload recruitable stroke work were significantly reduced in the animals that had both MI and MR. When trying to look at the molecular level, at the molecular cascades that are involved in remodeling, we looked first in collaboration with our good friend Roger Hajar, now at Mount Sinai, at the levels of the sarcoplasmic reticulum calcium ATPase. And as you can see here, the animals that had both an MI and a shunt had significantly lower levels of SERCA as compared with the animal that had only a myocardial infarction. And that applied to all areas of the myocardium, both at the border zone and in the remote zones. When looking at the remote zones, we saw that the pro-hypertrophic pathways as symbolized by the GP130 containing receptor, which is a receptor of the IL-6 family pathway, and the AKT, which is downstream for many growth factors and is a pro-hypertrophic pathway, are both upregulated at the first month follow-up, which is to signify that at this stage there is compensation. And we see that the remodeling is initially a beneficial thing, but they continue to be upregulated in the MI-only model, while in the animals that had MI and MR, they went down, signifying exhaustion of the pro-hypertrophic pathway, in parallel to persistent higher levels of caspase-3, which is the main center enzyme active in the apoptotic pathway. When looking at the extracellular matrix, and we looked specifically on the tissue metalloproteinases that are enzymes that are active in remodeling of the extracellular matrix, and I'm showing here only two examples, we saw that the tissue inhibitor of metalloproteinases, as signified by TIM4 in this graph, are significantly upregulated, but in parallel to non-downregulation of the matrix metalloproteinases themselves, signifying an active turnaround of the extracellular matrix. So to conclude this part, in this control model, moderate MR worsens post-MI remodeling, inducing larger LVs and reduced contractility. And while at first pro-hypertrophic pathways are upregulated, they subsequently fall and transformation to a failure phenotype occurs. And therefore, we concluded that MR can precipitate an earlier onset of dilated heart failure. Next, we proceeded to see whether repairing MR at this stage, at a very early stage, actually induces reverse remodeling. And sure enough, we can see here that we have three groups. We have the animals with the shunt, with only MI, with the shunt open, and animals in which the shunt has been closed by suturing it after 1 month. And we see that after suturing the shunt, we see reduction of volumes, both of the end diastolic, which is to be expected, but also of the end systolic, which symbolizes the improved contractility. And again, if you can see on the invasive contractility as measured by DP to DT and preload recruitable stroke work, you can see that repair induces reverse remodeling also on the functional level, and those hearts contract much better. At the molecular level, you can see that the repair actually induces a reversal of the situation here in the white bars on the left, the reversal of the downregulation of the GP130 and AKT, so they're upregulated at 3 months after repair, as we've seen in the animals that did not have mitral regurgitation in the first place. And we can see that the tissue inhibitors of matrix metalloproteinases were excessively upregulated, more than 4 times the baseline, signifying stabilization of the extracellular matrix in these animals. So we can conclude that repairing moderate ischemic MR after 1 month, early phase in the sheep, reverses ventricular remodeling at 3 months as compared to persistent MR, and that adverse molecular events that typify remodeling myocardium are suppressed after MR repair. The compensatory mechanism, including matrix stabilization, remain activated, and therefore repairing moderate MR early after myocardial infarction reverses the remodeling process exacerbated by mitral regurgitation. So this of course leads to the next question: what about later repair? If we can repair those animals at 3 months and follow them for 5 months, whether we would get the same beneficial effects. And we know that the long-term benefit, as Professor Schwamenthal has presented, the long-term benefits of repairing ischemic MR are not very good. So we tried to look at 12 sheep and followed for 5 months, as I said, and repairing them after 3 months. As then you can see here, we did not obtain any improvement in volumes and contraction in those sheep, notwithstanding the fact that they were repaired after 3 months, and contractility went down at 5 months in both groups, signifying that at this stage, repair was not beneficial. So we actually crossed the point of no return in repairing this valve. So, and this also reflected on the molecular pathways. There were no differences between the repaired and non-repaired animals. So in summary, the presence of mitral regurgitation after a myocardial infarction worsened prognosis, as we know from previous work. Mere moderate mitral regurgitation increases remodeling and worsens its impact after a myocardial infarction. Early repair, before failure phenotype has set in, permits reverse remodeling. Later repair is not associated with reverse remodeling in a controlled model. And the bottom line, as we all know, is that ischemic MR is a problem of the ventricle, at least in the beginning. So a robust ventricular solution for this ventricular problem is yet to be found. And I want to here acknowledge all the support that we got for this work. Both the Hadassah Cardiovascular Research Center team, in which the molecular part of this work has been done, the MGH Cardiac Physiology Lab, and again acknowledging Luis Guerrero is one of the most talented animal surgeons that I've had the pleasure to meet, and acknowledging also Mark Anschumacher, which is his software is the basis of all the work that we've done, and of course acknowledging Mori Verabi, Robert Levine, for all that I've got from him and I'm still getting from him. Thank you so much.